All right, we are recording. This is Cell Respiration Lab, BP Bio, Jones, Cell Respiration Lab. It works with 3 different chambers of respirometers. So, and you could see there's glass tubes, they're weighted. There's peas, Alaskan snow peas in one. Then there's cotton. And the cotton is actually of 2 types. On the bottom is cotton with soaked potassium hydroxide, KOH, in solution. And then above that cotton is a protectant cotton of just cotton. So it's just a non-absorbent cotton. So basically the cotton has KOH. All 3 have KOH, potassium hydroxide in all 3. KOH, 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 KOH. The potassium hydroxide is there for a very, very important reason. As you guys know, when anything performs cell respiration, what is produced? Anybody want to participate? I know you're on video. Anybody want to participate? Very good. Carbon dioxide. Carbon dioxide's made. And the problem with that is that carbon dioxide is made and oxygen's consumed. I think we know that if it's aerobic cell respiration. Well, we can't deal with that because oxygen's consumed, but KOH is made. Oxygen's consumed, KOH is made. Yikes, what are we measuring? So we have to cancel one of those, and we're going to cancel out the CO2. The KOH is going to combine with the CO2 to form a salt. Now I'm putting you guys in the spot. Does anybody know what KOH would combine? What KOH would combine with CO2 to produce? So in your brain, take KOH, add carbon dioxide in solution.
Oxygen gas.
Not oxygen gas. Not oxygen gas. We're not making more gas. KOH in solution plus CO2 in solution. CO2 in solution forms CO2
hydroxide, maybe?
Got it. Someone said it, the carbonate word. CO2 in solution forms carbonate and bicarbonate anion, or carbonic acid, which dissociates into bicarbonate anion and or carbonate and H+. So, okay, so now we have carbonic acid plus base, right? Carbonic acid plus KOH base, double replacement reaction. What's made? Carbonic acid plus KOH base, you get acid plus base, you get
Water and potassium bicarbonate.
Boom, there you go. Very good. Water and potassium bicarbonate, or actually potassium carbonate. But the point is, it's not so much the chemistry. The point is that we're grabbing the carbon dioxide that's made with the KOH. That's the point. We're taking CO2 out as a variable. Okay. So now you see germinating peas with this one. There's actually 25 exact peas here. I took those peas and I put them in this graduated cylinder. I did it this morning and I filled it to 50 mLs. Then I dropped them in, kerplunk, 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 one by one, and the water went to 60 mLs. So that tells me that the volume of these peas is 10 mLs. I did it by displacement. It was really easy. Again, I just filled this up to 50. I put the peas in here, and it went up 10. So that tells me this volume is 10 of the germinating peas. They're germinating Alaskan snow peas. So then I took 25 dry peas. There's 25 dry peas. And I put them in water for like 10 seconds. And the volume of 25 dry peas is not 10 mLs. So I had to add beads so that the volume is the same in both of these. Now I know it looks like the volume is different, but trust me, they're the same. This looks way less, but it's not actually. It's just because the surface area is so high here. There's a lot of space in between these. So essentially, there's 25 dry peas, 25 germinating peas, but they have equal volumes. And lastly, over here, we have X number of beads. I don't know how many it is. I don't care. What I do care is that it's 10 mLs of beads. I took water, I filled it to 50, I dropped the beads in, and it rose to 60. So that tells me that there's 10 milliliters of beads, or 10 cubic centimeters of beads. Basically, the same volume of beads that this displaces and this displaces. So each of these is the same volume. Isn't that nice? The same volume. I'm controlling for volume. I'm controlling for volume. Now you might be thinking, what are these beads, Jones, right? Well, those beads are glass. They're glass. They don't do anything. They don't perform self-respiration. Glass is not living, right? Glass isn't living, and that's okay. We're doing that to measure changes in temperature and pressure and compare those changes to this one and this one. We're going to eventually, not today, subtract out the changes we see in this vial from this vial, and this vial. So these are our experimental vials, and this is our negative control vial. These are our two experimental vials, and this is our negative control vial. Experimental. Negative control for temperature and pressure. All right, let's begin. Let's begin the lab. So I have a bath of water here, and the bath of water, I did it right before class started, is at 10 C. I don't know if you can see that, but it's at 10 centigrade. Period 1 did it at 25 centigrade. With you guys, I'm doing it at 10 centigrade. And we're going to just collaborate. I'm going to take these stoppers with 1 ml pipettes in them. The 1 ml pipettes are graded to 2 sig figs, meaning I can read them, like, for example, right there, it's 0.5, and then it's tenths of 0.5, so hundredths. So 0.51, 0.52, 0.53, 0.54, likewise. So I could tell to 2 sig figs, 0.01. I'm measuring to the hundredth place. 2 sig figs, 0.01 for each change. Sure, I could estimate the third, but I'm not going to. We'll just do it to 2 sig figs. And these are sticky, all goopy, but that's goopy for a reason. I gooped them up so that they go right here and they hold air or don't let water in. Now I blew these out to make sure that there's no water in here. Maybe look at these going, what are you going to do with those? Well, let me show you. Okay. I push these in nice and firmly. Nice and firmly. Okay. And the first thing I'm going to do is gently put them in this water bath so that the glass and the peas and the beads all equilibrate with temperature. I need the temperatures of these to be consistent, meaning I don't want any surprises. So I'm going to wait for 3 minutes and just wait for everything to get the same temperature. This is not time 0 yet. This is not time 0. Shreya is going to record for me the times. This is not time 0. This is the equilibration period. Okay, so we're not recording anything yet. I'm just, for another 2 minutes, I'm going to let everything in here equilibrate in temperature. See, I'm letting the glass, the rubber, the metal, this glass, the peas, the beads, the cotton, the KOH, all that stuff, all get to 10 C, because what we're measuring here is the changes in gas. The changes in gas in this 1 ml pipette. And if it's too hot in these chambers, what happens to gas? Does gas expand when it gets hot, or does gas contract? You guys know? Anybody?
Gas expands.
Totally, very good. Yeah, gas expands when you heat it. VP over T, volume times pressure over temperature. So if I warm these up too much, if I keep these too warm when I start, all the gas is going to shoot out. So I have to make sure that the gas outside in the atmosphere equilibrates with the gas inside these chambers. All the gas is all friendly. Now you may go, why do you care? How do you even know, Jones, right? How do you know that the gases are moving in or out? Aha! Food coloring. This is the best part of this lab. So we're gonna put food coloring, I call it a color load, into this chamber, into the pipette, excuse me. And the color should, if I do this right, it should move into the pipette. It should move in over time. Because peas consume oxygen and make CO2. But we're taking the CO2 out, right? The potassium hydroxide is reacting with the carbonic acid made by the CO2. So the CO2 is being taken out as a variable. And all that we're measuring is oxygen consumption by the peas, beads. I'll explain these in a minute, the beads. Okay. I think they're ready. I think I've rambled enough about that. Let's get to the fun part and color load these things. I'm gonna try to turn it so you guys can see. Actually, I'll do it like this. I think you guys can see like this. Hopefully you can see. This is the fun part. I really hope this works. It worked this morning. It took me a few minutes. It got a little messy. I don't normally get this messy, but let's try. Here we go. So this is food coloring. My recorder, Shreya, is standing by. I'll tell you when it's time zero, Shreya. Here we go. If I equilibrated it right, the color should get sucked in. Oh, come on. It's... There we go. That went in. Nice. That went in nicely. All right. I'll show you guys in a second. That one's not going in as well as I'd like. It looks like it is, but it's not. It's falling underneath. If I spill any, the water will be blue. These two did great. This last one, come on. Come on. This one just doesn't want to go in. There's no water in there. It did go in a little bit. I'm going to lower them and we're going to see. Here we go. Went in. Come on, go in. Come on. We'll blow this one out again. Technical difficulty. Hold on. Okay. There we go. All right. Beautiful. There was something clogging it. Done. Okay. Let me show you guys. All right. Here we go. Time zero. Time zero. Ready, Shreya? Germ.
Yeah,
You ready?
yeah.
All right. Germ. I'll show you guys. If you can see. Germ is 0.89. Dry, 0.91. Beads, 0.91.
Okay, got it.
Thank you. Germ is 0.89. Dry is 0.91. Beads is 0.91. Okay, so in 3 minutes, we're going to do it again. So right now it's 11:43. 11:46, we're going to take another reading. Set my timer. Okay. Clean up a little bit. It's like I didn't use red. It looked like a crime scene. Okay, so this is the experimental setup. You can see the germinating pea color load is already moving, guys. It's already moving inward. And the other two aren't really moving all that much at all. Everything is 100% submerged underwater. My water bath's at 10C. I don't know if you can see that. The water bath's at 10C. And everything's underwater. So while this is going on for the first time, let's briefly talk about what's going on here. The germinating peas, we got 2 minutes. The germ peas, oh, I brought them inside already. The germinating peas are plump. They're plump, and they're doing really well. The dry peas, they're not plump. They're dry. They're dormant. But are these dry ones cell respiring, right? Are they respiring? I don't know. Maybe they are, maybe they're not. I don't know. That's the whole point of this lab. The germinating peas should respire. Peas are like animals. They are like animals. They eat food in their endosperm, and they consume the food, endosperm, and they make from that endosperm carbon dioxide and water, like all cell respiration does. And they do it aerobically. Dry peas shouldn't do it at all. So the color for this shouldn't move all that much. It might move a little bit, but not all that much. Or maybe it does a little bit. Maybe they are somewhat active. Maybe. Now, the beads. We know why we have beads in the middle chamber. We have beads in the middle chamber with the dry peas to take up the same volume as the germinating peas. My germinating pea volume is 10 mLs. That's kind of irrelevant. But I make sure that the volume in the middle vial, in the dry pea vial, that's also 10. And then, of course, the number of beads at the end occupies the same volume as the other two. So all the volumes are the same. But why do we even have a bead one, right? Let's talk about that in a second. Let me take a reading for Shreya. Okay, Shreya, here we go. Ready? For germinating peas, 0.83. This is the 3 minute, right?
Yeah.
Yeah, 0.83. For dry, it's 0.92, and for beads, 0.92.
Okay.
Okay. Thank you.
Yeah.
All right. So in 3 more minutes, we're gonna do it again. That's 11:49. All right, guys. So what's up with these? Why do we do these at all, right? Oh, hold on. Why do these at all? Why do I have a vial just with glass beads? What's the point of
Because
that?
it's your control.
What's that?
It's a control.
It's your control.
Very good. It's a control. Excellent. Control for what?
The other 2 vials with the germinated peas and the dry ones.
That's right. It's a control for those. What are we controlling for, though? Right? What are we?
Changes in temperature or pressure that may affect the other beads the other peas as well.
Oh, yes. Very good. We are controlling for changes in volume caused by changes in temperature, VP over T, right? Because, look, I have this water bath on my workbench here. And I have it at 10C. I have a jug of warm water, and I have cold water down here. I have snow outside. See the snow? I got snow outside if I need to cool it down even. Freezer. Fantastic, right? However, it's 10C, right? But there's still little micro changes that occur in the temperature here and the movement of the water. The water's going to move. I'm going to bang into it. There's going to be slight differences in air pressure that's pushing on this water, right? Like Torricelli's mercury column. I don't know what is compressed, but air pushes on this liquid water, and that could change slightly the pressure in the water because the water moves and will move that color. So I have to account for that. And as you guys just said, I couldn't tell who said that. I'm sorry because I'm just looking at this video. But that blue beads only measures those changes in temperature and pressure. 49. All right, we're at 3 minutes. Next reading, here we go. Next reading, 0.79 for germ, Shreya, 0.79, 0.83 for dry, 0.93 for dry, and 0.92 for beads only.
Okay.
Thank you. All right, 49, 50, 51, 52, 52-ish, towards 53 actually. I'm a little late. But that beads only one, that's the blue one at the end, that's measuring changes in temperature and pressure that we cannot control. We can't account for them. We can't control for them. We can't take out temperature and pressure because we have to do this on the planet Earth, right? And there's changes in our atmosphere, which make micro changes here. So we have to subtract out all the readings from this blue one, the beads only. We can't reliably say any changes in this germinating vial are solely due to cell respiration. We have to take out any normal changes we see in the natural temperature change and pressure change, which therefore affect the volume in the beads only. So all the mathematical readings we take from the germ, we're going to subtract from those readings, those differences, these differences in the beads. The beads will tell us the differences. The beads are arguably the most important ones here. How are we doing on? Okay, we have 9 minutes. Can we do this in time? Let's see. What reading are we on?
So the next one is at 9 minutes. So that's like the fourth one.
You know what? We're going to go right to the bell. And I'm going to stop this at 12 and send you guys to lunch. And I'll hang out for 3 more minutes and do the last one.
Mr. Jones, would you like me to stay? Because I can stay if you'd like.
Oh, that'd be great. I'll just stop the video or whatever. It's not super important.
Okay.
That'd be cool. Yeah, we could stop. We can keep going. All right, we're about to take our 12-minute, right? No.
9 minutes.
Our 9. We're about to take our 9. So this is the part of the video where you may want to just fast forward. Or actually, you know what? I could probably pause the video here, right? Because it's going to be the same thing. Yeah, I'll just stop. Once again, to summarize, I'm going to stop the video. But this color, actually, we're going to take it now. I'll stop it. All right, Shreya, we're looking at 0.77 for germ, 0.77 for germ, 0.93 for dry, 0.92 for beads. Good?
Yeah, I got it.
All right, 55 we go, 55, 11:55. So I'm going to stop the video in a couple seconds, guys. Just to summarize, everybody out there who's watching, this lab has 6 of these vials. I'm only doing 3 just because I'm doing it from home in my garage. Period 1 did the 25C. Period 9-10 with me today. They're being great. They did the 10C. The one, but it's the same exact setup. Everything exactly the same. It's just that this is a 10C. Period 1 did it at 25C. There's 3 vials: germinating peas, dry peas and beads alone. The germ peas consume oxygen and make CO2, but we're taking this CO2 out. The dry peas are dry. They should do nothing, and it looks like they're doing almost nothing. And the beads alone obviously are not living. They don't make any CO2. They don't use any oxygen, right? But they do measure changes in temperature and pressure, which affect volume and therefore make the color move in and out. When we're all done here, we're going to take all of this data from the beads only and subtract it from the living pea vials. And we're going to get a corrected difference. And that's all in the lab. All right, guys, good job. I'll talk to you later. Farewell. Thank you, Period 910, for doing such a great job. I'm not saying goodbye to you yet. Hold on. I'm stopping the video. Stop.